The CRISPR-Cas system is a prokaryotic adaptive immune system that has been adapted in the laboratory for gene editing. In the lab, it consists of an enzyme called Cas9 bound to an engineered single guide RNA, or sgRNA, forming a Cas9 sgRNA complex. The role of this complex in gene editing is to make a double-strand break in the DNA of a target sequence. Cas9 contains two lobes, a rec lobe, involved in target recognition, and a nuc lobe, which has nuclease activity. The RNA also has two domains. At its 5' end, the sgRNA contains approximately 20 nucleotides that can be customized to complement the desired target site, followed by multiple stem loop structures at its 3' end, which are required for binding to the Cas9 nuclease. The sgRNA anneals to the complementary sequence in the target DNA, and the rec lobe of Cas9 binds to the duplex formed between the two. While the sgRNA recognizes the target sequence, Cas9 recognizes a short 3 or 4 nucleotide sequence of DNA known as the protospacer adjacent motif, or PAM. If the target DNA sequence complementary to the sgRNA is adjacent to a PAM, the Cas9 complex cleaves both target strands using the two nuclease domains in the nuc lobe. The structure of a Cas9 sgRNA complex reveals the two lobes of Cas9 and their association with the target DNA and single guide RNA. The rec lobe, in green, mediates the interaction with the sgRNA, in blue and red. The nuc lobe, in gray, has the two nuclease domains for cleaving both strands of the target DNA, in yellow. As the specificity of this nuclease is driven by a complementary sequence of RNA, the CRISPR-Cas system can be engineered to make a double-strand break in virtually any DNA sequence, provided it is adjacent to a PAM.